All right, here we are again, flipping the classroom. How appropriate. This is going to be the first lecture for Chapter 19. Chapter 19 talks about recombinant DNA technology or molecular techniques used in biotechnology. Okay. Recombinant DNA is, by definition, combining two DNAs from two distinct sources, essentially two different species. We can also call this genetic engineering. Okay, so we can use these techniques to analyze, alter, recombine virtually any DNA sequences from any number of sources. And so that's what we're going to talk about, at least partially, in this lecture. So the first technique we're going to talk about is DNA fingerprinting, because it hits on a whole bunch of molecular techniques that we need to know, and it makes a little more sense to me to show you guys and give you guys some of these molecular techniques with some relevance behind it. Our book kind of just lists a whole bunch of techniques without really bringing it together, and so putting it together this way seems to make more sense for students. It's difficult to understand some of these molecular techniques without actually doing them in the laboratory. We don't have a lab that goes to 360, so I'm going to do the best I can. So DNA fingerprinting is the name used to identify individuals based on differences in DNA sequences. So just like your fingerprints, your real fingerprints on your fingers are unique to an individual, so is our DNA. And so the process of DNA fingerprinting starts by first isolating DNA. And this can be from any source listed here. Whoa, sorry, not again. Alone. Ugh, honestly. All right. All of these types of fluids or body products. So anything that we can get DNA from, which needs to be a cell, some sort of cell that's somehow preserved. The first thing we need to do is called the polymerase chain reaction. The polymerase chain reaction's whole purpose is to amplify DNA, make more DNA, make lots of copies. Okay, and so PCR is essentially needs a DNA template, needs primers, needs DNTPs, some salts and other things, and a DNA polymerase. What does this sound, what physiological process does this sound just like? Now, if we think about molecular techniques, they're all based on some physiologic process. The one that PCR is based on is, yes, wait for it, DNA replication, right? DNA replication requires a DNA template. It requires primers. In the case of replication, they're RNA primers put on by primase. In the case of PCR, we use DNA primers that we manufacture or we make or order from a company. Okay, and then DNTPs, we know those are just deoxyribonucleotides. The reaction buffer are salts and other calcium salts, magnesium salts, other things that help the enzymes work, and DNA polymerase, just like DNA replication. So again, it's a DNA polymerase, which means it has to obey the rules. What are the rules of DNA polymerase? Yes, juicy 3' OHs can only add there. Everything has to be anti-parallel and complementary base pairing. Yes, my favorite things ever. All right, so primers are required to run PCR, to do PCR, to make polymerase chain reaction happen. Of course, this figure from the textbook, from our 107 textbook, is wrong, and I think this is an old version, so maybe the current version is better, but whatever. The region of DNA to be amplified by PCR or to be copied is actually from the end, the 5' end of one primer to the 5' end of the other primer. This would be the entire region that gets amplified. And so one of the primers needs to be complementary to one of the strands, right? And the other primer needs to be complementary to the other strand. So the primer sequence that they're showing here, up on top, that would be the 5' end of the primer, the 3' end of the primer, and it sits on this strand. This primer down here, this is the 5' end of the primer, this is the 3' end of the primer, and it sits or is complementary to that strand. Okay, and so it's complementary to that strand, and this picture's a little bit difficult to understand, but that's what the primers are like. Which means if it's complementary, what does the base that goes with G have to be? C, T, A, C, 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 A, T, etc., etc., etc. Complementary base pairing, right? Polymerase will grab onto the 3' end and start complementary base pairing to this template strand going that way and to this template strand going that way. Okay, so let's talk about the process. This is just a picture of the primers. So these figures are a little bit better. So first we have to start with our template DNA, right? So whatever we isolated, we pulled a piece of hair out of somebody's brush and it has that little skin tag, and we isolated DNA from that. We've synthesized our primers or ordered the ones. We have to know the sequence of the target in order to use PCR, right? The template DNA sequence, in order to make primers that are complementary to it. And then we need a whole bunch of nucleotides, deoxyribonucleotides. We first have to heat up the double-stranded DNA to separate it, right? Breaking hydrogen bonds, just like in replication, but in DNA replication, what does it? Helicase. In the case of PCR, we just use heat, so we don't need to use another enzyme, right? It's much simpler that way. So we have the single-stranded DNA, we have our primers in there, right? The next step would be primer annealing, and that just means complementary base pairing to the template. Complement to the template. And so when we go from really hot up here, and then we cool it down a little bit here, that allows these little primers to sit, or to a complementary base pair to their target sequence. Okay, and so if this strand here is 3' to 5', then this guy has to be 5' to 3'. We'd add to this end and it would fill in that way. 5' to 3'. When polymerase grabs on, it'll fill that way, adding to 3' ends, right? Always in the 3' direction. Okay, and so that's what they're showing here with these dark colored guys, right? That's extension. That's when DNA polymerase is active. Whatever temperature your DNA polymerase is most active at, that's where you'd set that temperature, and we are just doing complementary base pairing. So it's just like replication, right? Has to start with a primer, needs that juicy 3' OH, and then can extend. So here we go. These guys have extended. And then we just do this cycle over and over and over, which means we'd have to heat them. These two things would separate, right? And then the primer could sit back down on this template or sit on that guy. These two would separate. The primer could sit back on this guy or sit down over here on that guy, right? And we do this over and over and over and over. And if we repeat this 20 to 30 times, we can produce millions of copies of the DNA between, right, primer to primer. Okay, so all the way from that, and all the way from this, and then they'll reanneal coming back together as well. And that, my friends, is PCR. What they do in order to allow for this heating, right, when we did the beginning, when we heat to denature, to break the bonds, break hydrogen bonds, well, you can't, that's boiling, essentially, or almost, right? About 95 degrees Celsius, 100 degrees is boiling. So that breaks all the hydrogen bonds, but it also kills all enzymes, right? But Taq DNA polymerase is from a bacteria that's a thermophile, which means it's one of those bacteria that lives in the ocean floor hot sea vent where it's boiling water down there. So this enzyme does work at very high temperatures. So that way we can go through this cycle of heat, cool, extend, heat, cool, extend 20 to 30 times, and it does not kill our enzyme. So we don't have to add enzyme at every cycle like they used to have to. Woohoo, graduate students. And then here's just an overall better picture of the polymerase chain reaction. And if you only have a teeny tiny amount of DNA, you really only need a couple molecules in order for PCR to work. That's the power of PCR. So you can have a teeny tiny amount of the target, your primers, right? Add to 3' end, add to 3' end, denature, reanneal, extend here, right? Denature again, reanneal, extend, and so on and so on and so on, until you get millions of copies of the target sequence that occurs between, right, the two primers. That sequence right there. Okay, so here's another picture. Like all polymerase is just to remind you of the rules. Taq polymerase can only add to 3' end, 3' OH of an existing nucleotide, right? So if this is our template and this is our primer, there's our template. Our primer has to be 5' to 3', right? Has to be anti-parallel, and we're going to add to 3' ends. That means this guy has to be 5' to 3', this guy has to be 3' to 5', add to 3' ends. We'll get two identical DNA molecules formed, right? And we need to have that primer, just like in DNA replication. So be prepared for questions comparing PCR to replication. One of the main differences, well, PCR is done in a test tube and DNA replication is in a cell. DNA replication does all the DNA. The polymerase chain reaction only amplifies what's between two primers. The primers for DNA replication are RNA primers. The primers for polymerase chain reaction are DNA primers. We could make RNA primers, but we don't because it's more expensive and they're not very stable. So we just make DNA primers and it works just as well. All we need is that juicy 3' OH to make this happen. All right, now we're going to do some practice. So I'm going to walk you through how to design primers for PCR. And then in the next slide, you're going to do that for me and do a screenshot and an upload. So let's say we want to amplify the entire region shown. Where do we make our primers? That's question number 1. And then what is their sequence? Again, just like we did for DNA replication when we wanted to sit primers down at the origin, it was a little bit of trial and error. You can kind of do it in your head because you know we have to add to the 3' end. Right? Our primers need to go 5' to 3', and then this is the direction that we need to amplify. Right? So this little primer sequence can't be at the end and this falling off. We want to get all of this sequence between here and here, which means we'll want to sit a primer over on the left side, and we want to sit a primer over on the right side. So we can see that it needs to be, right, the rules, anti-parallel. And so if we want it to go in this direction on the left-hand side versus the right-hand side, right, we want our primer to sit going this way, right? So the first thing we're going to do is we're going to go 5' to 3', right? And then we can add, right, then we know we'll add in this direction. Okay, because we want to get this whole strand going this way. Doink, 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 right, over to there. And on this side, we know it has to be anti-parallel, and we have to add to 3' ends, so it needs to be going in that way, so we can go this way, right, doink, 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 all the way over to the end. So what does that mean? What does this line mean? These are deoxyribonucleotides. They need to sit down. When we say sit down or anneal, what do we mean? We mean complementary base pair. So this little stupid line needs to be complementary to its template. Okay? So what's complementary to T? A, G, C, T, A, T, C. And then polymerase, this would be the primer that we've made, right? If that's how big we made our little primer, here's our juicy 3' end. Polymerase would come in and continue complementary base pairing, right? G, C, 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 G, 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 T, 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 blah, 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 and so on in that direction. Does that make sense? That means our other primer must be complementary to this strand as the template. Right? Just like in DNA replication, both strands serve as a template strand for PCR and for DNA replication. So this guy would have to be G, G, C, G, A, T, T, A, C, G. And his 3' end sticking off of that, that would be the sequence of our little primer. And then Taq DNA polymerase would grab onto that juicy 3' OH and go G, T, A, C, G, G, etc., etc., etc. So what are their sequences? If we were going to write them out, and we always write things out 5' to 3', the left primer would be 5', A, G, C, T, A, T, C. And you only need to make 8 or 9 base pairs. And on the right, again, we always list them 5' to 3'. So this one's going this way. So we'd have to go 5', G, G, right? Because we're reading backwards. C, G, A, T, T, A, C. I guess I still had a G in there. 3'. That's how we always write out our primers. We never write them out 3' to 5'. That's just not conventionally how it's done. So you will be asked what is their sequence. You must write both of them 5' to 3'. The best thing to do is to do it exactly the way I showed you. Set it up on each of their templates. Write them out so that this one is 3' to 5' first. Then do the transcribe. Sorry, rewrite. I shouldn't use the word transcribe. Then rewrite this guy, right? This G is that G, G, C, G, A, yada, yada, yada. Is that clear? Great. Good job, people. Okay, now here's yours. This is the one that you're going to screenshot and upload to the PCR homework on Wednesday. We want to amplify the entire region shown. We want to add primers to the appropriate locations. Please label your polarities. Draw an arrow in the direction a DNA polymerase will extend. Okay? Add the primers to the appropriate locations. And when I mean that, not a line. Not lines. Use DNTPs. Okay? Use complementary base pairing. Do it exactly like you're supposed to. You can just write them next to the sequence. You don't have to rewrite them out 5' to 3' if you don't want to, but it would be really good practice if you did. Take a screenshot and upload. Okay, so after we've isolated the DNA and amplified it with PCR, and now for DNA fingerprinting, we amplify a bunch of regions of DNA on a whole lot of different chromosomes. Because if you just do one region, yeah, there's going to be people on this planet that have the same sequence in one region of one chromosome, right? But if you do 25 regions or 50 regions, nobody on the planet is going to have the exact same sequence. And so for DNA fingerprinting, to make it easier and to make it faster, instead of actually taking all those PCR fragments and sequencing them, we do something else to them that will show differences between individuals, and that's treating the DNA with a restriction enzyme. Restriction enzyme is just an enzyme isolated from a bacteria that cuts DNA. So cuts DNA or breaks phosphodiester bonds. Okay, in bacteria, these are used for defense against viruses, right? Viral defense. We were talking about having all those endonucleases or exonucleases that chew up DNA and chew up RNAs that are not protected. Well, bacteria make these restriction enzymes for the same reason. All different restriction enzymes cut at a different sequence, specific sequences, and so since everyone's DNA is a little bit different, when we amplify these big chunks on chromosomes, people's DNA will have different cut sites. This will result in different size fragments. So if everybody's PCR product started this big, right, somebody might cut here and get these two sizes. Another individual cuts there, right, and gets these two sizes. Somebody else cuts here and has 3 fragments. Okay, and that would occur at all 25 or 50 different locations. And so the pattern of all these different sizes of fragments is going to be unique to each individual. These different size fragments are just called RFLPs, or restriction fragment length polymorphisms, right? Poly meaning many, morphisms is what they look like or their shape, so their size. Okay, and how do we know all these different sizes? We have to separate DNA based on size, and that's called gel electrophoresis. And so when we're doing this RFLP analysis, what an example is their variable number tandem repeats, or vintners. These are also known as STRs, or satellite DNA. And so if we amplify satellite DNA can be found near centromeres and also in telomeres where there's just lots of repeated sequences, right? And so in this case, student number 1 or individual number 1 has 4 repeats on one chromosome and 5 on another, whereas somebody else has 6 and 3. When we cut these guys will have all different size fragments, right? These guys will have those 2 sizes, these guys will have these 2 sizes, and they will look different. If we do this at 25 to 50 different locations, and everybody has a different number of repeats, when we cut the DNA up, we will get those different size fragments I was talking about in the previous slide. All right, so gel electrophoresis is when we use an agarose gel, which is exactly like Jell-O gelatin, except that it doesn't have any sugar or flavoring in it or color. It's made out of agar, essentially, and it allows the separation of molecules of DNA based on size. So we put an electric current through that, and here's a little picture of the Jell-O stuff would be this stuff. There's little cutout holes in it where you can pipette in the DNA, right, or the restriction fragments that the PCR DNA that we just chopped up. And then we run an electrical current. We run to the positive from negative. Since DNA is overall negative charge, right? And you know our phosphate guy, remember that guy with all those oxygens and negative charges all over the place, which allows it to run to the positive cathode. So the DNA will travel down through the gel. Because it's Jell-O and it's thick, the big, large pieces will take longer to run than the small pieces. Small pieces can sneak through that big, thick, Jell-O-y stuff much better than the big, long pieces. DNA based on size. And so they call that DNA migration, right? So if the current is running this way, the DNA is getting pulled along with it to the positive, right? This would be the smallest piece, the next smallest, next, 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 and the largest. So the largest fragments are near the wells, or those little holes that we put it in. The smallest run towards the bottom of the gel. Okay? And then we stain these DNA fragments with that ethidium bromide I talked about in the mutations lecture. That's an intercalating agent that fluoresces under UV light. So you put this gel over UV light, and the DNA shows up, right, as all these little sized fragments. So these would be the smallest guys down here, bigger, 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 biggest. And then this is just a little setup. This is the apparatus where the gel is sitting right in here, right? This is the positive, this is the negative, and then this is the power source that we're running the electrical current through. And it has a lid on it so you can't stick your fingers in there and get zapped. Yow! Okay, and now we're going to take a little break because that was pretty tough, and we'll come back to it after this. Sorry that the resolution kind of sucks, but I think you'll get a kick out of this little deal anyways. Okay, nerdy fun. Hope you can see Lily up there. She's making a trail. Ah! I'm falling down now! Okay, I'm back on the trail. Here we go. Okay, I'm going to see if we could see. Oh, we can't see the trail. I was trying to make us a double helix in the snow. There it is.
Bye,
There it is. You can kind of see it. Right? It's supposed to be like this. Oh, wait, hang on. Here's my double helix. Right? Here's Lily's trail. She came down this way. And then here's my trail. Came down this way. And there you have it, people. A double helix. You gotta love it. You gotta love it. So what have we done so far? We've DNA fingerprinting. We did PCR, which amplified DNA. So we made a whole crap load. Oops, I can't spell. Right, and then we chopped it up. Right, that was a restriction enzyme. Right, and then we ran our gel. Right, we separated the fragments. Separate by size. But that's really all we know, right? We just know the sizes. We can't really tell what that DNA is, if we want to identify what bands are what, and not just here's a bunch of bands, we want to know what sequences belong to which chromosomes, especially if we want to compare pattern from one individual to another, we need to actually know the sequence. So now we're going to figure out the sequence of the fragments. And we're going to do it in a way that doesn't require DNA sequencing, which used to be really expensive, and now it's not that expensive. But this is still a faster, cheaper way to identify the sequence of the fragments. So that's exactly what we're going to do, identifying the fragments by something called southern blotting. So you can identify the sequence of the fragments by this method. So first we've separated all the fragments in the gel, and then we're going to transfer those fragments, that physically move that DNA out of the gel onto a nylon or nitrocellulose membrane, which then we can cause the DNA double helix to separate, break hydrogen bonds, so that we can add in a sequence that's complementary to those fragments that we can label with radioactivity. Okay, if we label them with radioactivity, they will glow, right, give off light, and we'll be able to detect them. And so if we want to know one specific fragment, we use a probe or a complementary sequence that is exactly like our template. And it will complementary base pair and identify where that sequence, where that fragment actually is on our. So we're going to walk through with some pictures. So here's our DNA fragments separated by gel electrophoresis, right? So we soak the gel in this solution so we can denature or break the double strands of DNA, right? Just the hydrogen bonds, not the phosphodiester bonds, right? And we stick the gel on top of this nitrocellulose or nylon membrane, right? So here's the membrane there, here's our gel underneath, and then here's that solution, okay? And then we have a whole bunch of like paper towels or blotting paper and something a little bit heavy, and we use capillary action for this liquid to soak up through the gel, through the membrane, into these paper towels, and that brings the DNA with it and sticks it onto that nitrocellulose membrane. So in this slide here, we have removed the DNA from the gel and have stuck it onto that nylon membrane. Now, it's not irreversibly stuck there at this point, right? But most of it is on the membrane. We still have a little bit on the gel, and we can take the membrane off, and then we treat that membrane either with heat or UV light to cross-link the DNA, actually physically attach the DNA to that membrane, which is why it can't just be a piece of paper. It has to be this special nitrocellulose or nylon membrane made just for this process. So the DNA on the membrane is fixed. That's what I was saying, UV or heat. And then it's stuck inside a little tube with some buffer and that radioactive probe, right? And so that's shown in red, and so the little radioactive probe is complementary to the DNA, and it should only sit down on the parts of the membrane where that fragment is. So if that fragment's here, and here, and here, and here, that's the only place that this little radioactive probe or this little radioactive piece of DNA will complementary base pair. And again, right, has to be anti-parallel, 5' to 3', 3' to 5'. So you generate this probe or this piece of DNA that you know is the sequence you're looking for, right? And it's single-stranded, and it's radioactively labeled. Again, you can use that P32. Remember way back when we used that radioactive phosphate to label DNA when we had the viruses in a blender experiment? Same idea, okay? And so whatever the sequence is, AGCT, right, that has to be complementary. And that's the only place that that probe or that complementary sequence that's radioactively labeled will sit down. And then those are incubated together at the right temperature for annealing and for everybody to be happy. All the crap is washed off, and what's left is just the places where the probe is actually hybridized. Hybridized meaning complementary base paired to its target. Autoradiography is where you put in a dark room this thing on a piece of film, right, and let it expose. Radioactivity gives off light, right? So every place this little band is, it's going to give off light. It's going to leave a dark mark on the film after you expose it and process it through the processor. And so that tells you that these, only in these places are those sequences that you were looking for. The rest of them were sequences that didn't match the probe that you used. And so what do they do? This is all together. This is the DNA fingerprinting in a nutshell. So we can put all of that together, right? They might collect a sample at the scene of a crime here, and then they have suspect number 1 and suspect number 2, right? They will do, collect the DNA, isolate the DNA, do PCR, right? Make a whole crap load of copies of that, right? And then they're going to run these on a gel, either cut them or not, depending on the protocol, right? Run those on a gel. One of the STRs, like I said, was the same idea as the vintners, the VNTRs. And then, right, they do the southern blot, then they'll put that guy onto, probe it with a radioactive probe that are complementary to the sequences we're looking for, and then compare the sequences of the sample from the lab to the different suspects. So, which I can't really see what they're doing here. Hang on. Of the identification from the 9/11 tragedy, because they wanted to return all the parts to the family members and not get any of that mixed up. So they used DNA fingerprinting to identify all those tissues. Okay, so let's take a look at this one. This is a little easier to see. In this case, they're looking at bone fragments, but we can imagine that if you were trying to look for, like we said, if this were the victim and then the rest of these people were suspects, you could try to see, you know, which ones, which, you know, tissues were from the victim and which were not. And so if this were the victim, right, and if this one were the fluids that were found that we think, you know, is from the perp, right, here's the perp, so that we'd know, so this is the victim, this was from the victim, and that was from the victim. So that was the victim's own tissues there, right? But any place else, this one, let's say, right, this was what was left at the scene, and this is suspect number 7. Well, look, his banding pattern matches exactly. And so this sample here definitely came from that individual. The other thing that DNA fingerprinting can be used, well, lots of things it can be used for, but one of the other main things they use for is to identify body parts and how they go together. And so number 1, you know, bone fragment number 1, 5, and 18 are all from the same individual. You could also look this 2, 7, I think just 2 and 7, right? Those are the only 2 guys that are the same. 4 and 11 have the same banding pattern, which means they're from the same individual. And so that's how they did. Okay, the other thing they can do is identify parents and children. So if this is mom, right here, look at this pedigree, yes, you remember this. And then there are 5 children, crazy, right? We can see that mom, right, gave that allele to each one of these, right? But this one here, right, and good old dad shared, right? Dad, mom, right? And so you can see how the siblings all have a different pattern, because you don't get the same exact egg and sperm unless you're an identical twin, right? Otherwise, you came from an egg from your mom and a sperm from your dad, but not the same exact ones as your siblings unless you have an identical twin. And so your siblings are all going to have, you're going to have similar banding patterns to some of your siblings, and you're going to have banding patterns similar to your parents, so about half from your mom and half from your dad. So you can figure out paternity and maternity using DNA fingerprinting, and also some relationships between individuals. 3 and 5 are obviously related based on similar banding patterns that they share. But you can see they don't share all of them, right? This one, and these are not shared between these two siblings, okay? But this one is shared with this one, right? But not that one. And so you can look at relationships among families using DNA fingerprinting as well. Here's another example. If you're looking at different adults and different children, you can figure out which adults are the parents of the children if you don't know, right? So there's 5 different adults here and 3 different kids. And again, using the same process, you can figure out that adult 1 and adult 3 are the parents of all 3 of these children, and that the other individuals were not. And that, my friends, is DNA fingerprinting. Thanks for listening, and I will see you soon.